Today I will be discussing the distinct roles for prefrontal dopamine D1 and D2 neurons in social hierarchy. Social hierarchy exists widely across many species and is a vital influence towards behavioral and social performance. Specifically, dominance hierarchy plays a role in accessing food, claiming habitats, and finding mates for reproduction. Those who are subordinate experience more social stress, which has an influence on physical and psychological stress. Recent studies have revealed that the medial prefrontal cortex plays a role in social dominance, as it is a region of the brain that supports social and cognitive functions. In humans, the prefrontal cortex is involved in both stable and unstable hierarchies. In mammals, the prefrontal cortex is involved with decision-making and social responses. In primates and rodents, social hierarchy is plastic and linked to agonistic interactions, such as aggressive social behaviors between males. Once established, the hierarchy is relatively stable, preventing excessive aggressive behaviors among group mates. At the cellular level, the strength of excitatory transmission in the medial prefrontal cortex is one of the critical determinants of inter-male hierarchy, as bidirectional manipulation of synaptic strength switch the dominance relationship between males. The medial prefrontal cortex has been implicated in diverse aspects of social behaviors, including encoding of social cues, social recognition processing, and adaptive social coping. It is speculated that the medial prefrontal cortex manages to process all these different types of social behaviors through parallel neuronal microcircuits. Prefrontal dopamine, or DA, represents an attractive target for neural influence over various social, cognitive, and executive functions. Dopamine plays a role in encoding stimuli, which underlie enhanced avoidance and defensive behaviors in animals who are susceptible to social stress. Prefrontal dopamine modifies the functions of the prefrontal cortex, mainly through the D1 and D2 receptors. D1 and D2 receptor expressing neurons in deep layers of the prefrontal cortex exhibit distinct electrophysiological properties, morphologic characteristics, and projection targets. Because of the different intracellular signaling pathways mediated by the D1 and D2 receptors, dopamine exerts divergent actions in synaptic transmission and plasticity. Activation of these neurons plays a critical role in social information processing in rodents. However, it is unknown whether it plays a role in social hierarchy regulation. Here, we characterize the physiological properties of D1 and D2 expressing prefrontal neurons from dominant and subordinate mice and explore their contributions to social dominance. The question that is being answered today is, do dopamine receptors contribute to social hierarchy regulation? Male mice who are 6 to 8 weeks of age were first housed in a group setting with controlled environment and equal access to food and water. During the experimental period, the mice were co-housed. To measure dominance hierarchies in the group-housed mice, the food reward-based tube test was conducted. Group-housed mice were placed on a food-restricted diet consisting of 2 to 3 grams of food per day for a week in order to keep 85 to 90% body weight before restriction of food took place. A rectangle chamber was divided into 2 compartments and was connected by a transparent plexiglass, as seen in Figure A. During the habituation process, mice were given 2 days for habituation in the chambers. On day 1, mice were subjected to exploration for 5 minutes in the chamber, and frosted Cheerios were placed in the chamber as a reward to encourage exploration. On day 2, mice were placed one at a time in the chamber, and the reward was only placed at each end of the tube. During the training session, on day 3 and 4, the mice were trained for 8 consecutive trials to run through the tube and consume the food reward at the end of the tube. During the testing session, on day 5, mice were subjected to walk through the tube and consume 3 Cheerios just like the training sessions before they participated in the test trials. During the test trial, 2 cage mates were placed in the tube on opposite ends and met in the middle. The one that caused the other to retreat received a greater dominance score. This can be seen in Figure A above, where the dominant mouse is seen to the right and the subordinate mouse is seen to the left. The social rank of the mice was determined once the win-lose relationship between a pair of mice was consecutive for 3 days. There was no difference in body weight among the pair of mice. Coronal slices of the medial prefrontal cortex were created after a social hierarchy was determined from the experiments. Whole cell current clamp recording was used to measure basic electrophysiological properties of layer 5 D1 and D2 pyramidal neurons in the prelimbic region of the medial prefrontal cortex. It was found that mice who were dominant had a larger amount of postsynaptic response from D1 receptors, and those who were subordinate displayed more activity in their D2 receptors. Overall, it is seen that dominance is not only a characteristic gained through natural selection. It is the activity of neurons present within an individual of a species determines whether or not they will possess dominance. To characterize synaptic properties of different dopamine receptor-expressing neurons with respect to social rank, slice recording from pairs of group-house transgenic mice expressing red fluorescent proteins under the control of the D1 or D2 receptor promoter were observed. It was found that the amplitude, but not the frequency, of MEPSCs were significantly larger in D1 neurons from dominant mice compared with subordinates. This suggests that a robust postsynaptic efficiency of D1 neurons is associated with the dominance hierarchy. In contrast, it was observed neither difference in the amplitude nor frequency of MEPSCs between dominants and subordinates from the D2 mice. It was found that the AMPA-NMDA ratio was significantly higher only in D1 neurons from dominants. Overall, the results suggest that the more robust glutamate transmission selectivity at D1 receptor neurons is associated with social dominance. The findings revealed, furthermore, that prefrontal D1 and D2 neurons play distinct but synergistic roles in social dominance. Enhanced excitatory synaptic transmission at D1 neurons is associated with dominance, and activity in prefrontal D1 receptor-expressing neurons is required to maintain high rank status. In contrast, hyperactive D2 neurons are associated with submissive status, and manipulating the D2 neuronal excitability does not affect social rank in either dominant or submissive mice. However, simultaneous manipulation of D1 neurons in dominant mice and D2 neurons in subordinate mice is sufficient to switch their dominant-subordinate relationship. The data indicates that activity in the D1 and D2 neuronal subpopulation in the medial prefrontal cortex regulates social hierarchy dynamics in dominant and subordinate mice. So far, the results suggest that enhanced synaptic strength of D1 neurons in the medial prefrontal cortex is critical for social dominance, and the hyperactivity of D2 neurons is involved in submissive status. In addition, it is found that inactivation of the D1 prefrontal neurons caused dominant mice to adopt a subordinate social rank. The inhibition of D2 neurons did not affect social rank status in dominant or subordinate mice. In the prefrontal D1 neuron inhibition experiment, it was found that inactivation of D1 neurons in dominance caused a significant drop of social rank. Importantly, the dominant D1 mice remained in high rank when receiving saline treatment. In contrast, the inhibition of D1 cells of subordinate mice did not cause any rank changes in the rank status subordinate mice. It was found that silencing prefrontal D2 neurons affect neither dominant nor subordinate status. The findings of this experiment highlight how social conflict recruits specific neuronal circuits and plays a role in establishing social hierarchy. Social rank changes are also positively correlated with manipulated medial prefrontal cortex activity. It was seen in subordinates that there was hyperactivity in D2 neurons, and in those animals who dominate, there is hyperactivity in D1 neurons. In conclusion, the findings provide novel insight into the divergent functions of prefrontal D1 and D2 neurons in social dominance. The D1 and D2 neurons within the medial prefrontal cortex work synergistically to regulate social hierarchy. Critiques and additional applications of this experiment would be to see whether or not the way that a species is raised influences the dopamine neurons and their status in a social hierarchy, or if social dominance is determined strictly by an individual on its own. In order to do this, mice would be raised in a high-stress environment and a low-stress environment. It is hypothesized that those who are raised in a high-stress environment will exhibit greater activity in dopamine D1 receptor neurons